Okay, so hello to everyone. Good to see you. I'm Monica Paule, the CEO of gene editing company Cazyme, and also a board member at Lithuanian Biotech Association. And I'm very honored and at the same time very happy to invite you today to the scientific symposium, Gene Editing from Research to Industry. And we have this symposium on a really great day. Just a couple days ago, we had like the World CRISPR Day that we celebrate each and every year. So a great timing and the great world of gene editing we all be talking today about. So first of all, I would like to thank our partners and sponsors. So first of all, I would like to thank the main sponsor, Thermo Fisher Scientific. So please give a round of applause. And it's also great coincidence that we have this symposium at the same day as Thermo Fisher Scientific Days, which happen each and every year. So really a day that we have a lot of interesting presentations and discussions about the most novel things in science. I would also like to thank CASIM, which I'm also representing, also Lithuanian Research Council and Vilnius City Innovation Industrial Park, and also our partners, Go Vilnius with their new brand Vilnius Tech Fusion, and Life Science Center, who are hosting us here today, and well, which is the real international, worldwide known hub of life sciences and gene editing as well. And also I would like to thank my colleagues from Lithuanian Biotechnology Association, who all participated and actively contributing to organizing this event. So please give one more round of applause to all the sponsors and partners and organizers. Okay, so actually I'm not the one who's opening this symposium. And I would like to introduce, well, I would say the main person why we are all here, who actually started all gene editing here in Lithuania, but actually contributed greatly and made a significant impact to the gene editing as is today. So I would like to introduce you Professor Virginijus Šikšnys, which definitely all of you know. And Professor Virginijus Šikšnys currently holds, well, a chair of distinguished professor here at Vilnius University. He also serves as a chief scientist and head of department at the Institute of Biotechnology here at Life Sciences Center, Vilnius University, and is the chairman of the board at the center. He's also a founder and the chairman of the board at gene editing company CASIM. And Professor Virginijus Šikšnys' research on the CRISPR-Cas has had a major impact in the field of gene editing. And he, together with co-authors, published semifinal papers of Cas9 biochemistry that were the foundation of the translation of CRISPR-Cas bacterial immune system into powerful gene editing tool. He's a member of European Molecular Biology Organization, also European Academy of Microbiology, as well as Lithuanian Academy of Sciences and Norwegian Academy of Science. His work has been really recognized by various awards and prizes, including very well-known Kavli Prize. So now, please give a big round of applause to Professor Šikšnys.
Thank you, Monica. Thank you, Monica, and it's my great pleasure to be here and welcome you at the Gene Editing Symposium that is held today at Life Science Center. And this symposium is a kind of satellite meeting of a big meeting of European Federations of Industrial Biotechnology that will start tomorrow in Vilnius. And this EFIB meeting actually brought to Vilnius a big crowd of top-level experts in industrial biotechnology. And I'm really glad that gene editing is also discussed within this framework of EFIB meeting. And the CRISPR-Cas genome editing technology was discovered 10 years ago, and it actually contributed to very different fields of biotechnology, and currently it's used for precision gene editing in plants and animals, and also engineering of industrial microorganisms. And the broad limit of the broad range of applications of gene editing technology is now probably limited to our collective imagination. But of course, you may ask actually the relevant question, why specifically genome editing, and why CRISPR, and why Vilnius? And I'm always proud to show this map that I borrowed from the review that Eric Lander published in 2016 in a journal of Cell. And Eric called this map the map of major CRISPR discoveries. And I'm really glad to find Vilnius on this map. And if you look at the timeline of CRISPR, so I think that, ooh, so we don't see slides. Interesting. Sorry about that. Oh, okay, so I'll get to the map by that was published in a review of Eric Lander. And as I said, I'm really happy to find Vilnius on this map as one of the major players in the CRISPR field. And if you look at the timeline of CRISPR, so I think the year 2012 is a really important year, because at this year, both we and other teams published a seminal paper that described that the biochemistry of Cas9, and in our paper we claim that actually Cas9 could be genome editing. And if you look at this timeline, so the time in the timeline of CRISPR before 2012, we can call it BC or before Cas9, and the time actually after 2012 we can call AC or after Cas9. And in fact, the CRISPR started as bacterial antiviral defense system that was probably first identified in late 90s. But then before 2012, the CRISPR-Cas systems were considered as antiviral defense systems. But after 2012, actually, they are more often called like genome editing tools, especially when we talk about Cas9. So, but in my research career, BC, before CRISPR and before Cas9, actually, we were studying another antiviral defense system that is called restriction modification system. And actually, at that time, we were aimed to understand actually how restriction enzymes that act as DNA scissors are able to discriminate their targets. And actually, when we were doing our research on restriction enzymes, in parallel, the industrial biotechnology was developing also in Vilnius, and many of you probably still remember the company called Fermentas, and Fermentas was worldwide known distributor of restriction enzymes and other enzymes for molecular biology. And then, at some point, Thermo Fisher acquired Fermentas, and now actually Thermo Fisher is an important industrial biology player here in Vilnius. So in our research, AC, after Cas9, we still continued to deliver and published together with our team a couple of important papers that were focused on development of novel genome editing tools. And I would say that at some extent the history here again repeats itself, because when we were studying novel genome editing tools, a company Kazime was established in Vilnius that now delivers all these genome editing tools for a research community. So I wouldn't like to take much of your time today, because actually we have really an outstanding line of speakers, and I would like to thank you all for taking part in this genome editing symposium, and looking forward for the presentations. Thank you.
And yeah, small presents for our speakers. Okay, so the second presenter will be online. Sending regards from beautiful California, and very sorry for not being able to come here. So she's Dr. Kiana Aran, already well-known person here in Lithuania, and Kiana sends her all best regards because she unfortunately had to be on a different event at the same time, which will be soon publicly announced, what kind of award she got. And I would like to present Kiana, who actually has a lot of relations with Lithuania, and is doing research collaborations both with Professor and Kazime, and contributed greatly to a Women in Biotech initiative that we launched several years ago. So Kiana Aran is an assistant professor of medical diagnostics and therapeutics at Keck Graduate Institute in California, and is a visiting assistant professor at University of California, Berkeley. She's co-founder of Nanosense Innovations and co-founder and the Chief Scientific Officer of EuropaBio in San Diego, California. Dr. Aran also serves as a drug and delivery medical diagnostics consultant for the Bill and Melinda Gates Foundation. So Dr. Aran obtained her PhD at Rutgers University in New Jersey and completed postdoctoral work at the University of California, Berkeley, and the Buck Institute of Research and Aging. Dr. Aran's primary research focus is on combining the power of biology with electronic transistors to create biology-gated transistors on chips. She developed the first CRISPR-powered transistor, termed CRISPR chip, that is capable of searching for targeted regions of the genome in real time using CRISPR as the search engine, and the ultra-sensitivity of graphene transistors to report the search result. CRISPR chip was the most read paper in Nature Biomedical Engineering in 2019, featured on the front cover, and was featured over 50 media news outlets. Kiana Aran now is working on expanding the technology beyond CRISPR, and she's a great example how different disciplines can interact and bring great scientific and also applied results. So please, let's see Kiana's presentation.
Thank you very much for inviting me for this symposium. I was really looking forward to being able to attend in person and meet the people that I have been working with over the past few years in person. And unfortunately, due to multiple overlapping events, I was not able to make it in person, but I have recorded my presentation to you, and I really hope that you enjoy it. So as I was introduced, my name is Kiana Aran. I'm an associate professor of bioengineering at Keck Graduate Institute. I'm also the co-founder and chief scientific officer at EuropaBio, a biotechnology company that develops electronic to be integrated with biology. I'm also the co-founder of CRISPR QC, where we develop electronic analytical tool for CRISPR quality control. And I'm also a visiting professor at University of California, Berkeley. Today, I want to talk about the integration of CRISPR and nanoelectronics, specifically nanotransistors, and how this integration and this combination have allowed us to have a few applications that you otherwise could not have. So inside your computer, there is a CPU. CPU is basically is a platform that processes all of the function in your computer. There's also a GPU that takes care of all of the graphics in your computer. In CADEA and in my lab, we are developing BPUs. These are the next generation of biosignal processing unit that can really connect with biology, that can connect the live biosignals to the digital world. So these BPUs consist of multiple different components. The major component of these BPUs is transistors. So before I dive more in depth into how this BPU basically works and how we combine it with CRISPR, I want to give you a brief history of transistors. So in early 1900s, we had vacuum tubes. So vacuum tubes are basically used mostly as electronic switches. And they were used for telecommunication, in radios, and even I remember that my grandfather still owned the radio that used vacuum tubes. But for long distance telecommunication, they were not very feasible because they would break, they would require maintenance, they would often explode, they would consume a lot of energy, and they get overheated. So around 1950s, it was when AT&T commissioned Bell Lab to actually develop better switches, something that is better than the vacuum tube, something that could overcome the challenges that vacuum tube had. So this is when William Shockley and John Bardeen and their colleagues basically developed transistors. So transistors are, again, electronic switches that are more compact, they consume less energy, and the ability to basically do it and develop this transistors was switching to material from vacuum to germanium and silicon. And this was basically when the whole era of silicon, semiconductor was basically created. So just switching from vacuum to germanium and silicon, which later on silicon become the more dominant material, enabled us to actually create this more compact transistors. So developing transistor was really advancing. Then we were able to actually compact the whole component of a system that allowed us to basically create integrated circuit. These are things that the circuit and system that you see inside CPUs, inside GPUs. And really, if it was not because of the invention of transistors, we would never have such compact laptops, phones, and today we can really go to much smaller size. However, the generation of this transistor enabled us to really advance in terms of electronic gadgets. But now if you want to use all of these electronic gadgets to something that can connect with our biology, then there are some challenges. So this is where my team in the company and in the lab are developing this next generation of transistors that are made from a different material. So we are switching from silicon and germanium to graphene now. Graphene is 2D material that is biocompatible, that can actually connect us with biology without impacting biology or without biology impacting it. So this is when we have developed this BPU. These are biosignal processing units that have transistors that are made from graphene. So again, I want to reemphasize that switching from vacuum to germanium and silicon, or to silicon, really enabled us to have so many different gadgets, so many different applications. And today you can really not imagine the world that we live today without transistors. Now we're switching by switching the material from silicon to graphene. This is a quantum material, it's a 2D material to enable us to have application beyond what we are capable of doing today. The invention of transistors and integrated circuit and graphene, these are all Nobel Prize winning technologies. And we really, what we do in Kadia, we're basically putting all of this together to create the next generation of these transistors. Why do we use graphene instead of silicon? Because graphene is biocompatible, the most important parameter. It's a single layer of carbon molecules, and it's super fast. That means that it can conduct much faster, meaning that when biological events are happening, which every single biological event is an electronic event. These are charge-charge interaction in our biomolecules. Those interactions can be recorded and can be sensed much faster than silicon, because silicon is a 3D material, is bulky, so the signal will dissipate before we can detect it. And this will really allow us to have a time resolution in terms of like watching the biological event that semiconductors such as silicon cannot give us. So this is the major point of using graphene for our transistors. So as I mentioned, each BPU has multiple different components. The transistors, which we call it the nanoware, is only one component of these BPUs. We have to develop the biology. Basically, we call it the wetware. This is the biology that sits on these transistors. We have the reader that actually can read the signal from these transistors, and also analyzer and software that can analyze this signal that we're observing, because these signals are not very easy to handle. They're very complex, and we have to understand the biophysical properties of these molecules in order for us to be able to understand these biological events. How does this transistor function? Basically, if you look at the picture on the left, this is basically a schematic of a graphene that is sitting between two terminals. We immobilize a capture molecule. This capture molecule can be an antibody, can be an enzyme on the surface of the graphene, and this is a functionalized transistor now. And this functionalized transistor have a specific electronic signal that we call the base signal, basically. As we expose these transistors, and this is the picture that you see on the right, as we expose this transistor to biofluid or a sample that contains that analyte, the interaction between the analyte and here we're showing an antibody and antibody can change the conductivity of this graphene material, which is very sensitive to charge-charge interaction, or anytime a charged molecule comes close to it or charge atop this transistor changes, we can sense that. So that results in changing conductivity of the transistor, therefore changing the overall electrical parameter of these transistors, and this we can detect in real time. So this is basically the overall function of these transistors. So here what you see on the left is the actual picture of one of our BPUs. If you zoom into the middle part, you see there are these black rectangles. And each of these are transistors. If you zoom in each of those transistors, you can see the graphene layer and the analyte and the capture molecule that is, the capture molecule is immobilized on the graphene, and then you have the whole biology happening there. There are 15 transistors on this specific design, and but there are 3 circuits. That means each 5 transistor basically report together. And if one of them was broken or something happens to the transistor during the process, we don't lose the signal. So we can immobilize many different molecules. Sorry, we can immobilize many different molecules to these graphene transistors. You can have an enzyme, you can have an antibody, you can have an antigen, you can have a peptide, and basically anything that interacts with your analyte can be immobilized and conjugated with these graphene transistors. And the reason that it's easier to conjugate this material with graphene is that graphene is carbon basically bond, so it's much easier for us to actually find linker molecule that binds that biology to something that is sort of made from biology. Our whole body is made from carbon, and we can actually link these two together. So as you can see, you can monitor antibody-antigen reaction, antigen-antibody reaction, a small molecule antibody reaction, enzyme as it binds to its analytes, and many other biological interactions. Because we can immobilize many different capture molecules on these transistors, you can imagine that you can use them for cancer diagnostics, detecting exosome. You can have an antibody that binds to a surface exosome marker. You can detect an immune response. You can detect a circulating tumor cell. So it makes this system really a multi-omic system. And by only changing the capture moieties, you can basically detect different type of analyte without a fluorescence label. This is really important because one of the major challenges that we have with fluorescence labels is that as you're trying to expand this analytes, then the fluorescence molecule and the processes really changes from each of these analytes. So it makes it really, really difficult to actually have a system that could monitor all of these analytes together. But because we don't need that labeling, and because we can really scale to hundreds or thousands of transistors, and each of these transistors can potentially detect one analyte without any label, you basically get a signal from each of these analytes. And let's not forget that when transistors were first invented, we had like 1 or 2 transistors, but within 10, 20 years, we went from 1 to 2 transistors per chip to 70, 80, 100,000 transistors, per chip. And now we can really scale those up. We're not there yet, but then because this process has been done in the past, there is a path for us to move forward to scale the number, more than 15 to the hundreds or 1,000 potentially, and do measure all of these analytes together. So now let's go to how we actually combine CRISPR with these transistors. We have done many, many analytes, but for this talk, I'm just going to focus on how we combine CRISPR with these transistors and what application we were able to use it for. So back in 2017, when I was still a postdoc at UC Berkeley, 2016-17, I was fascinated by CRISPR. To me, CRISPR was this fascinating enzyme, a fascinating robot that could unzip double-stranded DNA, search for its target, and once it found this target, then it could, cut or cleave or do, and then later we could do the editing. So without the search function of CRISPR, basically the CRISPR molecule had to basically do the search function first before we can do the editing. So then I thought about, this is an amazing robot. It can basically detect target sequences in the cells. Can we actually utilize it on the chip and detect target cells, target sequences on the chip using CRISPR? So that was the whole idea. So this is exactly what we did. I'm going to show you this video now. This is what we did, but it took us a few years to basically develop this, but the outcome was amazing. So if you zoom into the middle part of the chip that I showed you, there are again 15 transistors. Now, if we zoom into each of these transistors, we anchored a CRISPR molecule to the surface of the graphene. Of course, we had to go through a lot of process to actually understand how was the best way to anchor this CRISPR molecule to graphene without impacting its activity, basically not hindering its active site. And once we did that, when the CRISPR molecule actually was exposed to the DNA molecule, this is, when we talk about DNA, this is whole genome. This is not an amplified sequence. It could basically do the function that it was doing in the cell. And if it found its target based on the RNA molecule that we have, programmed it to detect, if it finds the target, then it could bind to the target, and then we could monitor this binding. Because DNA is a very, very charged molecule. So if the compound binds to the DNA molecule, then we could easily detect it. For the first application, we used the activated Cas9. This is a Cas9 version that basically binds to the target sequence, but does not cut it. And we wanted to do that because we wanted to make sure that we have a good signal. Although Cas9 also works, because when Cas9 binds and cuts, it still remains bound to a part of the DNA in the absence of the cellular molecules that could basically kick it off. And we're still able to detect it. So the first application that we did using CRISPR chip was basically, and we call the technology CRISPR chip, it was basically a chip and a CRISPR. We published our first result in Nature Biomedical Engineering in 2019. It was the cover of the journal. It was one of the most read paper. It was the most read paper on that year, and it was the first time that CRISPR molecule was basically integrated with electronics. After that, there was a lot of sensor being reported that were using CRISPR as a sensing mechanism, but we were basically the pioneer in doing that application. And the beauty of that was because our chips are super sensitive, we were able to actually do DNA detection, target DNA detection without amplification, which was, I think, a game changer. Because over the past 40 years, we have been doing PCR, where we basically take the genome, chop it off, then we have to amplify it, then label it, and then we were able to basically detect the target sequence. We basically run over the sample by bus before we're able to detect it. But we really took away all of those processes, used CRISPR, used biology as technology to basically eliminate the PCR process or the amplification process in our detection. So we did a lot of testing, but this is one of the testing that I want to present here. So the whole idea, as I mentioned before, was that imagine having a chip that has hundreds of transistors. If each of those transistors are functionalized to detect a specific sequence in your target genome, then you can potentially have a chip that could do targeted sequencing for 100 targeted genes. So this was really the idea of, can we have yes, no, is there a mutation here? Yes, no, if this gene exists, yes, no, and basically have this whole array of transistors that can report to us which genes are present or not by just changing the guide RNA that we use in our CRISPR enzyme atop these transistors. To prove the concept, we actually worked with a few different genes from HEC-BFPs to other type of genetic materials. And we also did some clinical testing in Duchenne muscular dystrophy. So the plot that you see here in the middle basically was the result from CRISPR chip that was designed to detect exon 51 in the dystrophin gene. So Duchenne muscular dystrophy can happen due to mutation in all 79 exons of the dystrophin gene. The most common are exon 51, exon 3. So those are the ones that we selected because we could get the sample for them. So sample H1, H2, H3, A, B, C, they all had exon 51, and sample D, E, and F, they were lacking exon 51. So as you see, we were able to actually detect the presence or absence of this exon on that sequence in exon using our technology. And the plot on the right shows that we can also, be quantifiable. This is really amazing because quantifying with PCR is not a very easy process because you're amplifying the sequence, so much that sometimes you basically, there's no way for you to quantify it, or it's very difficult and it requires a lot of control to be able to do that. Because we're not doing amplification, we can easily quantify how much of that sequence or how much of that gene we have. So that's another capability of our platform, which basically it's really putting our platform ahead of many other existing platforms. So again, this was the whole idea. We developed another CRISPR chip that was detecting exon 3. We have reported that in our paper as well. So here in the table, you see on the bottom, we're showing that we can detect the presence or absence of exon 3 and exon 51, but we could potentially do that for all of the 79 exons and really show yes, no, the gene is there or is not there. So it's basically an amazing platform for genotyping. We developed, so then we started the company, we developed the first technology that was using CRISPR on transistors. We call the system the genome sensor. Basically, you could genotype easily with our sensor using CRISPR. And the technology won many, many awards since its inception. But we did not stop here because every time we pitched our technology, people were asking us, okay, you've done a great job with detecting deletion, large deletion, insertion, but can you actually do single point mutation? Because 50% of genetic diseases, human genetic diseases, are the result of single point mutation. So in collaboration with Dr. Virginidis and of course, Dr. Virginidis Sickness and Calcime, we were able to actually complete and continue this work where we showed that with our sensor and with selection of the right Cas enzyme, we can even detect zygosity, we can even detect single point mutation without amplification. So the zygosity was a bit difficult, and it's, I think for this specific sample, we were able to do it. I'm not sure we can do it for all the sample, but for this specific sample, we were able to do it. So we were able to do, if you look at the plots, the first plot on the right, basically the middle one, we were able to detect single point mutation in sickle cell disease, where we have only a single point mutation, and this we were also able to detect with our system, and we were also able to detect a zygosity, and this was also really amazing to be able to do. And we also expanded the sample that we were testing to ALS as well, because ALS is also a single point mutation, is a result of single point mutation. So we were also able to detect the presence of single point mutation in those samples using our CRISPR chip technology without amplification. So this work was also published in Nature Biomedical Engineering as a continuation of that work, which really further showed the capability of our platform. This table is really showing the performance of comparing the performance of CRISPR chip with other technology that came before or after us. As you see, the CRISPR chip technology is the only technology that does not require amplification. Every single technology out there still requires amplification. And we were able to eliminate amplification by just using CRISPR biology, or by using biology as technology. We further collaborated with some, agricultural companies, and we were able to actually detect the presence or absence of transgenes in plants without amplification. And this is really important because when you look at the application, when you have the infrastructure, it's easy to do, genotyping. But when you're, for example, in the field and you want to do genotyping really quickly, then it becomes PCR and amplification is really not feasible. It's something that basically the technicians or the lab technicians in those industry really do not want to do it, because it just requires a lot of work, and they may not do it that often, and they really more using like lateral flow assays for those applications. But if they really want to do molecular testing and genotyping, they have to do PCR, which is very difficult right now. So this platform can really offers a great tool for those farmers or for that industry to be able to do genotyping in plants. So that the graph that you see here, we were able to detect the presence or absence of 2 genotype from wild type without amplification, which was also a great application of our technology. But one of the great things about our technology, and I talked about, as things start binding and unbinding to our transistors, you can monitor those activities in real time. So this, what you see here is basically the process of how we functionalize our chip for CRISPR application specifically. So you have, let me just see if I can turn on the pointers easier, do it this way. So here you have your bare graphene, and then you add a linker molecule. The linker molecule binds to graphene from one side, and it has a different chemistry on the other side, and can be anchored to Cas protein, in this case, deactivated Cas9 or dCas9. After we do that, we basically passivate the rest of the surface, so we don't have non-specific interaction. Then we introduce the guide RNA to form the complex. We have done a lot of studies on how to put the complex. Should we form the RNP first and then put it? But this way, putting the enzyme first and putting the guide RNA later was the process that gave us the most reliable result. So at this point, the CRISPR chip is ready to be tested for genotyping and being exposed to genomic DNA. But the interesting part is that what we did not realize is that this final stage where you have your guide binding to the Cas protein itself was a product. And this we really started realizing it after talking with customers. And this is a really important, aspect that I want to point out here, that as an academic at the time, if I wanted to actually go and just, try to push the technology for genotyping, I would have never realized that there is other market application for this technology. And it's really by collaborating with industry, the right industry, people who are actually working on this technology, is the only way that you can find other potential that could be beneficial for your application. And I remember once, we were working with a team member with Dr. Virginia Sickness, and my lab could not get the technology to work. We had a Cas enzyme, specific Cas enzyme, and we were trying to actually see if we could monitor the function of that Cas enzyme on our chip. And my grad student was not able to get it to work. And eventually we realized that my grad student was using a wrong one component of the buffer was not added to that to her buffer. And it was amazing to be able to actually see that you won't get the exact same result if you change one component, and we could see the reduced activity by just changing one buffer component in our chip, which also showed a capability of our chip being able to detect if the condition is not the right condition, or if you have altered the condition somehow that is now not corresponding to the original performance that you were expecting. The same thing with guide RNAs and Cas. When you talk with people who are using CRISPR, many of them are assuming that when the Cas and the guides have bound together, they form a very stable complex, and then there is no way for these two to dissociate. And that's not true, because these are electrostatic interaction. And sometimes if you change the pH of your system, then you might be basically dissociating the guide, or changing any condition of your system. You might be dissociating your guide from the Cas enzyme, and that will result in the function or in that you were not expecting, or reduce or alteration in the function of your CRISPR platform. So as I was saying, that it's our platform can monitor all of these activities in real time. So if you look at the way that we functionalize our chip, this is the signal that you get from our system. You basically can see as you're binding the Cas enzyme, as the guide RNA is binding to the Cas molecule, and all of this can be monitored in real time. So now, if there is any alteration in the guide RNA in the environment, for example, the pH, or in your Cas, and as we know, there's a lot of companies that are developing many different Cas orthologs for different purposes. and their function and their characteristic compared to the original Cas enzyme or whatever Cas enzyme that you're trying to compare with can be really monitored and evaluated with our system. So this really led to starting up the other company, because we realized that this was really important for biologists to understand, how the alteration in environmental condition, in the guide sequence, in the target sequence really altered the function of the RMP, the CRISPR complex, and how that would impact the outcome. So we started the CRISPR QC company, which was basically solely focusing on partners, commercial partners, that what they wanted to get insight to these activities and improve their design. So basically, the system can play a role where for companies that are doing nucleus engineering, what type of a Cas enzyme they're using, if they're using a different Cas and Cas9 for various different reasons, for example, for temperature stability, for improved, performance, and they want to compare it with the original Cas enzyme, they can basically compare the two in our platform together. The design of the guide, how does adding chemical moieties to the guide for different purposes can impact and alter the binding of the guide RNA to the Cas enzyme, which then potentially alters the function of this complex, because if the guide is not completely binding to the pocket of the Cas enzyme, then the Cas cannot change its conformation the way it's supposed to do it, and it may not cut precisely. So all of this can be something that we can monitor with our platform. So basically, CRISPR QC, we're not saying that CRISPR QC can answer all of the questions, but there are many questions that only this platform can answer that could potentially be helpful. So now CRISPR QC is working with many companies that are developing basically different type of Cas enzyme, different variant of Cas enzyme, and they want to compare their activity in terms of binding or cleavage, and we can do that with our platform. So this is a really easy way to compare those activities without having to go through the whole process. This is an example of just comparing the quality of the proteins that you're receiving from different vendors, because there is no rigorous quality control for this vendor, and this shows you that when we selected 3 different vendors to get our Cas, and this is not even different Cas ortholog, these are the same Cas enzyme from different vendors or different batches, and you can show differences. You can see differences in the way they perform. So that's something that we can definitely monitor. The other thing is that you can monitor the performance of the activity at different pH, which is really important, because what is your storage buffer? Where are you delivering your Cas, your CRISPR complex? What is the pH condition? Is it always stable? Are you changing the pH condition? And you want to select a complex that is more stable at different pH condition. Here we were working with one of our partners where they had different guide RNAs, and we monitored their we challenged them with different pH, and we looked at to see which one of those complexes basically changed behavior more than the others. And as you see in this list, some of the high binder, high performer remained high performers throughout the whole activity. Basically, the darker blue line shows that the high performer remains high performer as we change and alter their pH. But some of the guides, for example, some of the guides basically they altered their functions. For example, the GC-rich guides, they were behaving differently in pH 4.5 than pH 6 and pH 7.45, or the relay guide RNA was performing much better in pH 6 and 7.5, and then was declining. Its activity was declining in pH 4.5. So can we actually develop some sort of an analytical tool to be able to give those answers to the customers? Of course, this will require us also to correlate this result to the outcome, to the cell outcome, to see what is the correlation between this instability between the guide and the Cas enzyme and the final outcome. In addition, we can monitor the kinetics of this binding cleavage. And this is super, super important because as there's many industries that are moving forward to using multiplex guide RNA to do multiple editing or using multiplex guides for different purposes, these guides, especially if they're expressed in the cells, these guides perform or basically compete to bind to the Cas enzyme. So now if you're separately monitoring the cleavage and activities of one RMP, and then comparing it to the others, and then combining it and sending it to the cells, or expressing those guides in the cells, are you actually expecting the same outcome? The answer is no, because some of these guides basically outcompete the other guides to bind to the Cas9, or other Cas enzymes. So you basically will not get, will not form those RMP to start with to do any editing. So really understanding that if you had a combo of guide RNAs together, how do the combo basically interact with the Cas when they are in abundance, or which guide RNA basically outperform and binds more strongly to the Cas enzyme. So those we can monitor with monitoring the kinetic of how these guides bind, and then challenge them with introducing the second guide and see if they get kicked out. So those sort of things, these are things that we can do with our platform. So right now, as I mentioned, it's really, really important that we, as we're developing these analytical tools, we also monitor to see how these results from our analytical tools correlate with the cell outcome. So this is something that is super important for us. And right now, we're collaborating with NIST, Syntigo, and we had funding from NIH and CRISPR QC. It's supporting this studies where we can basically correlate this analytical studies to the cell outcome, and we want to see, can we actually utilize these parameters without the need to do the follow-on cell experiment to identify and select the best complexes, the best guide RNA, the best Cas enzyme, the best condition, and be able to improve our cell editing outcome. And one of the major things that we also do in our in our companies is that we really remove the human factors, because these studies are really, really important, not only for the purpose of identifying the best complexes, but also really understanding how complexes work. What are the changes in the guide RNA that have the most profound impact on the editing outcome, or what are the changes in the cells that could basically correlate with some of these analytical studies that could basically result in better or worse outcomes. So those are not really understood, and there's a lot of data, but because each lab is basically developing their own data, they have their own processes, it's not easy to use that data to understand to understand CRISPR better. So we're here. I'm showing you a video of our platform, basically where you have multiple different chips. Everything is automated. There is no user error here. And so basically, we're trying to minimize the errors that are introduced to the system to be able to get results that are more, cohesive, and then we can really trust the outcome. And of course, NIST is also playing a major role in trying to unify all of these people who are working in the CRISPR field to follow the same process. So I think together with having analytical tools that are automated, with having people and scientists that are following the same processes, we'll be able to understand the CRISPR technology much better for editing application. And I finally want to talk about, take 1 minute and talk about the alliance between industry, academia, and government, and how this collaboration can be really a game changer for science and economic future. Because with, for example, now I talked about NIST, we're working with NIST, with Syntigo, CRISPR QC, us from academia, and we're really trying together to develop this analytical tool that could really push forward the boundary that, or the potential limitation that this technology could have, and try to understand them early on so we really can scale it and increase its safety for gene editing application. It does require academia to do the basic science, because we have more resources to do the basic science due to the access that we have in universities, with the government to fund those activities, and with industry to really guide the academia and government to what is it that actually needed to scale this technology to make it safe, and these are really required. My lab has been extremely collaborative with industry and government, my academic lab, and only through this collaboration I found my research to be more useful. So we're also developing the next generation of transistors that can monitor really in real time the conformational changes in these enzymes, and this is something that I'm hoping to have within the next few years for you guys, where we can use this electronic system to monitor as the enzyme is opening up, closing up, and this is something that we're working on right now, and this has been funded by the government and the industry to basically develop this next gen. So this alliance is really required for us to actually push the technology's boundaries. With that, I want to conclude and acknowledge my lab member in my academic lab. Without them, none of this work was possible. And my team at Kadia, these are the people who are actually doing the work. And it's been amazing to have such an outstanding team. Also, our collaborators from all across the globe. This work is all collaboration. And I have not presented, a lot of things that we have been doing with this transistor and with BPUs from, different type of multi-analyte that we have been detecting from collaboration with other companies and government, and without this collaboration and this fundings, none of this work would have been possible. So with that, I want to thank you all, and I really hope that you guys enjoy the symposium, and I hope that I can join next year. Thank you very much.
Okay, now it's working. So thank you all for the applause for Kiana. She will be definitely watching the recording because it's currently very early for her, like a nighttime. So she will be really grateful. And I had a chance actually to visit both Dr. Kiana Ran laboratory at KGI Institute, and also to see how these CRISPR chips are produced in EuropaBio and CRISPR QC, and that's really, really impressive. So I hope that you found this presentation really interesting and valuable. Okay. And proceeding with our program and presentations. So next, we will have a guest from Thermo Fisher Scientific, Dr. Jonathan D. Chestnut. And Dr. Chestnut received a PhD in cell and molecular biology from UC Davis and did postdoctoral studies at the University of Colorado in recombinant antibody technology development. Prior to graduate school, he was employed at the Salk Institute and Hybrid Tech Inc. in La Jolla, California. After his postdoctoral research at Colorado, he joined Invitrogen as a research scientist. Since then, he has led the development of various prokaryotic and mammalian cells cloning and expression systems. He has led groups focused on development of topo and gateway cloning, as well as the human embryonic cell engineering technologies. He currently leads a team in the cell biology business unit that is focused on cell and gene therapy platform development. Their efforts span the workflow from immune cell culture, cell processing, payload delivery, genome editing, and the final processing on the therapeutic cells. So please give a round of applause to Dr. Chestnut.
Thank you. It's a pleasure to be here today. Let's see if I can make this work. Okay, so I'm based in Carlsbad, California. It's great to be here in Vilnius. We work closely with the site here in Vilnius, so I try to make it out here as often as I can. I'll take a short trip through kind of how my team is focused with respect to genome editing tools for cell therapy. And so I start with this slide to talk about the cells that we're focusing on engineering. So really two types of cells. I think if you follow the literature, you've heard of T-cell therapy, engineering T cells with chimeric antigen receptors to direct them toward tumors for killing. And part of the engineering is inserting that CAR construct into a specific locus or randomly, depending on the delivery tool, and also knocking out various inhibitory receptors. And initially and currently, that type of therapy is focused toward what's called autologous therapy. So that's where if I'm ill, I take a bag of blood from my vein. The T cells are isolated. They're then engineered. They're allowed to expand, and then some weeks later, they're re-injected into me for the therapeutic effect. Two things about that make it non-desirable, other than it actually does work, but the vein-to-vein time there can be up to a month, and the cost is quite high. So the therapy itself is not as accessible as one would like. So the push in the field is to move down toward the bottom here. Is allogenic therapy. So that's something where you would create a cell line that's engineered toward a particular tumor or transformed cell, and then those cells would be kind of an off-the-shelf therapy. So if someone is diagnosed, they can go into the clinic, and there would be a therapy there right away. The cost should be significantly less, and there should be little or no waiting time. So for CAR cells, again, patient donors, for pushing toward allogeneic therapy, we're working with natural killer cells. They're slightly different. Obviously, they're a different cell. Those cells are naturally directed toward non-self antigens. So they recognize in your body as part of your innate immune system, identify things that you've been infected with, and they kill them. So we can either try to use those as native NK cells, or more and more we're trying to engineer those with chimeric antigen receptors. And then currently a lot of the source of those cells are from healthy donors. And then the ideal goal for those is to be able to engineer iPS cells with the appropriate CARs, and then differentiate those cells into NKs and be able to freeze those down for a real allogeneic therapy. So that's the road that we're trying to focus on. So here's the workflow, really for both allogeneic and for autologous therapy. First, there's what's called a leukapheresis when they draw the blood out of the arm. And then there's a selection and isolation of T cells. And so we recently launched this instrument on the lower left called the CTS Rotea. That's a counterflow centrifugation system to where we can isolate very pure T cells out of patient blood samples in a very short period of time. Then those cells are generally activated. So we have a Dynabead product that will activate those into really activated, ready-to-go T cells. And then what we want to do is insert the CAR construct. So up until recently, a lot of that engineering has been done using lentiviruses with a packaged CAR construct in them. Those are very active. You can integrate a lot of CAR constructs into cells. The integration is not specific, so it goes everywhere in the genome. While that does work, it's not considered that safe in the sense that it could activate oncogenes, and then the cells tend to not be regulated as one would hope. So we're really pushing toward using tools like CRISPR and talons, which I'll touch on, to insert those CAR constructs into a specific locus. The favorite locus in T cells is the T cell receptor locus, so we can knock out the T cell receptor, the endogenous T cell receptor, and replace that with a CAR construct. So the feeling in the field is that now that CAR construct is being more naturally regulated in the cells, and those cells are more robust. And then those cells are then, the engineered cells are then expanded and then cleaned up and then reinfused into the patient. Similarly, the allogeneic workflow was we would do that and then freeze down the bags and then be ready for therapy. So, as I kind of mentioned, currently lentivirus is used to put the CAR constructs into T cells. Relatively safe, but there's a lot of holdbacks from that. What we're trying to push toward, the field is also using now, is either CRISPR as an RNP, a ribonuclear protein complex, and then using donor DNA that's packaged in an adeno-associated virus. Non-integrating virus, but it's very efficient in delivering the single-stranded donor DNA. What is the next step toward that is going to a non-viral delivery platform. So using, it says here plasmid DNA, but we use more and more linear single or double-strand DNA as the donors, and either CRISPR or in some cases TALEN. And so that gets us to kind of a more specific integration. We do not have the viral integration all over the genome. And then finally, trying to get away from the electroporation piece in the middle. So we're mostly using electroporation to deliver those nucleic acids and the RNPs to a specific lipid nanoparticle delivery. And so that's something that the cells would be less speed up, they would be higher viability, which would get us more cells for the therapeutic application sooner. So we're somewhere in the middle right now. So I'll talk kind of about in the middle today. A little bit more on the Rotea system, just a small tabletop system that's able to process a leukopak, that's a bag of cells from a patient very quickly. And it basically can isolate the cells. It then can clean them up, wash them, and concentrate them to whatever concentration you want. And then it can pump it out into a different bag for the activation or the other steps. It's also used down in the workflow as well after engineering to clean up all the different components from the engineering piece as well. So I talked a little bit about electroporation. I think if we're using mammalian cells or even bacterial cells, we'd use electroporators, very small scale. What we're aiming for, especially in autologous workflows, to be able to deliver payload to large volumes of cells. And so what we recently launched is the Xenon instrument, and this is a high volume electroporator. So we can either do a single shot, which is a 1 ml cartridge. So 1 ml is the smallest this instrument goes right now, or the multi-shot, which is what we're aiming for the clinical workflow. It will do 25 milliliters of concentrated cells. And it will deliver our payload, either the CRISPR or the TALEN RNPs, or the DNA donors in the same mix. So let's talk a little bit more about engineering. So I mentioned lentiviruses. There's current limitations on these. So to package the viruses, it's very expensive. The DNA needs to be made at a cGMP grade, which is quite expensive. There's another cost driver, and that is the DNase to clean it up afterwards. So a very high cost for doing that. Again, there's safety concerns about where that virus integrates in the genome. You're limited on insert side of the lentivirus, maybe about 6-7 kb. So CARs fit in that nicely because they're only about 3 to 4 kb, but if you want to do anything more more elaborate or add anything at multiple CARs, you tend to run out of size. Again, we're not inserting these constructs into specific loci, so they're not necessarily regulated as we would like. So as the push toward the gene editing approaches, we have other things that we need to worry about. So we worry about the efficiency of knocking in our CAR into a specific locus. We want that as high as possible. We want to be able to knock out the inhibitory receptors. So there's 4 or 5 of those that we want to knock out. And we want to make sure that they're as specific as possible. So we want to reduce or eliminate off-target effects. So if I'm cutting somewhere, does it cut somewhere else in the genome? And insert size is another question, right? So if we want to put in a CAR, that's generally doable. But if we want to put in more elaborate things like new operons or other sorts of things, it's difficult to get large pieces of DNA into the cells very efficiently. And back to the knockout and the knock-in. Ideally, we could do that all in one shot, right? So we could knock out our TCR, T-cell receptor, knock in the CAR at the same locus, and knock out the other inhibitory receptors in the same reaction. That would be ideal. But what happens in a percentage of those cells is you get chromosomal damage and the effect of deletions and chromosomal translocations, et cetera. And that's a real thing. And so we do measure that. And the other piece is if you're going to commercialization, there's IP landscape, which we don't really like to think about, but it's true. CRISPR has its own IP surrounding it, so one would have to have the appropriate licensing to be able to commercialize. So back kind of the basics. I think we all know this, but I just wanted to run through this anyway. For a genome editing tool, we want to have something that cleaves with a high efficiency and a very high specificity. Most of the current tools are double-strand cleavers. There's new tools out there like base editors and prime editing, which you're seeing in the literature, and those are becoming more and more popular for sure. So they're very applicable. But in this case, what I'm talking about, these are I'll say the old-fashioned double-strand break cleavers. And again, either you can do a knockout and that DNA is fixed by non-homologous end joining, or you can add a piece of DNA, or the cell will add the piece of DNA by HDR, homologous directed repair. And so taking the balance of those two pathways, in some cases, we will use a small molecule inhibitor to push maybe from the NHEJ pathway to favor the HDR pathway to increase the efficiency of insertion. And the tools we all know, CRISPR, so my team, BC, working a lot on TALENs and integrases to do cell engineering. And then AC, we pivoted a lot to CRISPR, but we did maintain some programs on TALENs, and I'll talk a little bit about that, kind of where we've gotten with the TALENs as another tool for genome editing as well. So, transition from the bench is critical. So a lot of us are doing basic research, so we're interested in getting a tool that works very well, that we can get quickly, and that is really accessible. CRISPR is, by a great stretch, the best tool for that. But as we get into the clinic, we want to make sure that we take our research tools and they're made at a quality level that we can go directly all the way to the clinic, in through clinical trials and commercialization. So quality is very critical. The mount, quality, depending on how large of a lot you're using. For these 25 milliliter processing experiments, we use a lot of donor DNA, we use a lot of guide RNA, we use a lot of CRISPR protein. And so we have to make sure that we have the right quantity and we can scale. And then finally, all the reagents that we're using in the buffers need to be of cGMP quality. So through years of hard work with the team here in Vilnius, we have launched last year a cGMP grade Cas9 protein. And so you can see some of the attributes of this enzyme. So basically, has very high performance, which I'll go through in the next few slides. They're sold currently in 2.5 or 5 milligram vials. Again, I'll go through a little bit about the cGMP traceability and documentation. It's a significant amount of work. If you haven't been involved in cGMP processing or development, it's quite a learning experience. The manufacturing is done here in Vilnius by a very highly trained and well facilitated facility over here that uses fully closed system to create this enzyme at very high quality. And these done are in very large lots. So generally we're getting about 20 grams of purified proteins in one pass. And again, we're using maybe a few hundred nanograms for a single reaction, so it's definitely a lot of reactions that we can get. So some of the testing that goes into a cGMP kind of clinical grade enzyme, and these are all documented assays. So the purity has to be high, greater than 95% by various different measurements, less than 5% of any kind of detectable aggregate. We have to check the identity of the protein various different ways, and then look at process-related impurities like DNase and RNase and host cell protein, host cell DNA. Of course, we can't have any real amounts of endotoxin, has to be sterile, et cetera. So there's a lot of testing that goes into creating a very high-quality grade enzyme, much more than you would see with a research grade. I don't know where that slide came from. Okay. So let's talk a little bit about performance. So this is a flow cytometry diagram of an experiment we did where we used CRISPR to knock in a CAR construct. I'm going to see if I can get the pointer to work. Oh, good. So here is without any Cas9 or CAR construct. And this CAR construct we're using is an AAV CAR donor, so you'll see it's very high efficiency. So here's basically a TCR positive, CAR negative cell population. And then, of course, if we still do not add Cas9, but we add the AAV CAR, we're not getting insertion of the AAV CAR, the population stays negative. If we add Cas9 without the CAR, we see about a 96% knockout of the CAR, of the TCR, T-cell receptor. So we're getting a very high efficiency knockout of the receptor in those patient-derived T cells. And then if we add our AAV CAR, now we're getting about a 90% knock-in of that CAR construct at the T-cell receptor. So very, very efficient creation of a CAR T-cell here. And then if we do a CAR T killing assay where we basically mix these engineered cells with a tumor cell line, and then we can measure the ability of these cells to actually kill those cells, you can see that we get a very high efficiency killing of these cells. So it's very efficient. We can do this in high volume. So these are cells that would be really ready for the clinic, although they still need to have the other receptors knocked out as well. I'm going to take a little bit and talk about our new TALEN platform. So TALEN does essentially the same thing that CRISPR does. This is a plant pathogen protein that binds certain sequences in the DNA, and normally in nature it delivers transcriptional activators. in the plants. Several groups have found that we can engineer these proteins to deliver various things to the DNA. In this case, we're delivering a nuclease to the DNA, and we're delivering half of a dimeric complex. So you can design them such that they bind either side of the place where you want to cut, and then the nuclease will form the dimer and cut the DNA. The historical TALEN that you'll see will have a FOC1 nuclease. Our team has identified and engineered a novel nuclease that we think increases the activity for sure. And then here is kind of the comparison between our TAL platform and the Cas9 platform. What is going on? Sorry? 2, 3. One more, okay, don't touch that. Okay, so in this case, we can see our CRISPR knockout here without any donor is still 96. The tau nuclease is performing identically almost. And then when we add our CAR donor, we're seeing similar insertion. And then when we do the killing assay, these cells perform exactly the same. So the point here is that there are other tools in the toolbox that we can use to create T cells, CAR T cells, and depending on the commercial appetite for doing specific licenses, one can use one or the other of these tools. Okay, so I want to talk a little bit about off-targets. So in the initial days of engineering mammalian cells with CRISPR and with TALENs, the thought was, that it wasn't very specific and that people weren't really asking the question as much. And then we and others started to develop tools to be able to identify and map off-target sites in the genomes. And so I think as we get further down into the clinical application of genome editing tools, having off-target mapping as a lot release assay is going to be very important. So we really use kind of 3 different platforms here to look at off-targets. So if you notice a guide RNA, some guide RNAs are very specific and some aren't very specific. Some guides are very active at a particular locus. Some guides lead to a complex that's not very active, so it's kind of a balance. There are different algorithms to look at a certain guide sequence and try to predict off-targets, and those are generally a good guide. Pardon the pun. But also, there's other sequencing tools out there to be able to identify the actual off-target sites. So basically, what we use, we call it tag-seq. It's our adaptation of guide-seq that you might have seen in the literature. And so basically, that technique is when you put in your cleavage reagent, CRISPR. We also put in a blunt double-strand DNA tag. And when the double-strand break is made and when it's closed, that tag gets trapped. And then we can use that to prime for doing sequencing, and then we could map where these off-target sites are. And that gives us a set of loci, and then we can go in and confirm that using what we call targeted amplicon sequencing. And so we can develop a panel of, basically take all the potential off-target sites that you identify in your next generation sequencing. Could be a lot, could be hundreds, and we can create a panel of amplicon sequencing for that to be able to check and confirm the off-targets. The other thing that I mentioned is basically we can get transpositions when we're doing multiplexed. And so in this is a simple diagram. We can get a transposition of different chromosome ends. We can get large deletions of chromosomes that are hard to pick up using these other techniques. We can get transversions, et cetera. So in this case, we want to be able to identify those as well. And so we're adapting different tools to be able to identify and map those to be able to qualify the reagents that we're going forward with. So what do we want to do? So in the end, when we're getting to the clinic for these clinical tools, we want to make sure that we have the highest activity tool we can get as long with the highest specificity. So we found that delivery of, for CRISPR especially, delivery of the Cas9 as a protein, as a preformed ribonuclear protein complex is the most efficient because you get the cleavage activity in the cell, and then that protein's degraded rather quickly. So then the CRISPR complex isn't sitting around trying to find something to do. Then we want to go to our highest scoring guide RNA and then be able to follow up with that with our different analytical tools to be able to identify that we have had a good specific reaction. And then what I haven't talked too much about is going toward these high fidelity Cas9s. So we've taken the wild type Cas9 and we've mutated it into our what we call our high fidelity Cas9, and that's in process up the street here, and we hope to have the cGMP grade of our high fidelity Cas9 out by early next year. So getting to the point where we have the most specific and active tools possible. So really, this is my last slide. I think the goal here is to get to the best enzyme, highly active and highly specific. We think our wild-type Cas9 or wild-type Cas9s are very active, but not terribly specific. We think our high-fi Cas9 of the ones we've tested against is the best. And we are still doing the analytics on our talons. We predict that our talons are at least as good as one of the best Cas9s. So we hope it's somewhere in that circle going forward. And so the goal is to have the highest quality, the most active, and the most specific tools to be able to get to the clinic in the safest way possible. So I'll just end right there and be happy to take some questions.
So, any questions?
Yeah.
We need...
Is there a microphone?
Yeah.
Is it on? Yeah? Okay. Thank you for your talk. It's a little bit ironic because, I mean, people nowadays, in the Western countries, when they go to the shop, they look for the food, should be no GMO food. And we will actually coming now to the 21st century where we're creating GMO people actually, where if you become, getting this therapy, so you eventually will be like genetically modified in a way. And I was wondering, when you're using these technologies, I think one of the drawbacks of the initial application of the CAR-T was that for some people you develop actually autoantibodies, which basically reduces the effect of the CAR-T cells in the patients. Not in all, but in some. So I wonder whether actually the technologies which you provide will overcome some of these limitations, which in the long term could lead to the better outcome. Can you comment on that?
Oh, that's a great question. I think I would imagine the autoantibodies would be directed toward the CAR construct. Yeah, and we do not work specifically on creating novel CARs or single-chain antibodies or that sort of thing. But in general, I think having the cleanest tool, the most specific tool to be delivered to the patient would reduce that. But I don't. And that's a really good point. I don't know I don't know how we would do that unless you would engineer extracellular domains for the CARs. That would be the most minimally antigenic as possible. But yeah, that's a really good point.
Any additional questions? Okay,
All right.
so thank you very much for a great presentation.
Thank you.
So now we have a half an hour coffee break, just outside the auditorium, and very much looking forward to see you back here at 3. Okay, so welcome back. Good to see you after the coffee break. And we have still 3 very interesting presentations about gene editing in different fields. So I really think you will learn a lot, and it will be a very interesting couple of hours in front of us. So we will start from Dr. Giedrius Gasiūnas, who is the Chief Scientific Officer at gene editing company CASIME, and he's responsible for leading all the scientific and research activities. So Giedrius also is a senior researcher at Institute of Biotechnology and at Life Sciences Center, Vilnius University, and member of Young Academy at Lithuanian Academy of Sciences. He obtained PhD in biochemistry at Professor Šikšny's laboratory at Institute of Biotechnology, Vilnius University in 2012. During the PhD, he studied the mechanism of Cas9 protein activity, demonstrating that it is the programmable RNA-guided DNA endonuclease. Giedrius Gasiūnas is a co-author of 28 scientific publications, which are cited more than 3,000 times, and was involved in several Cas9-related patent applications. So, welcome, Giedrius, and a round of applause.
Okay, so first of all, thank you for kind introduction. And today I would like to share a few stories about research activities illustrating research activities we are performing in CASIME. And usually I start from slides introducing our company, but since you hear a lot about CASIME today, so I will skip a few slides and save some time, hope. Maybe I only shortly mention that our main activities and fields are providing research services for CRISPR application, exploring new Cas proteins for various applications, and also developing and characterizing new CRISPR-based molecular tools for various industries. And our main competences, like CRISPR biology, knowledge of CRISPR biology and applications, also genome editing tools and their application, cloning expression and purification of proteins, and in vitro protein RNA characterization allows us to achieve these goals. So you also have heard about gene editing a lot today, but I will remind shortly that for genome editing, the tools which introduces double-stranded DNA breaks in specific places of genomic DNA are required, and then these double-stranded DNA breaks are repaired by cellular machinery to get desired changes at exact position. And it's difficult to overestimate the importance of this technology, because it's already did a lot for research. Also, it's becoming more and more important for medicine, for example, for curing genetic disorders and creating new anti-cancer therapies, and also in agri-sciences for advanced plant breeding. So, and the most popular tool for genome editing currently is so-called protein Cas9, which actually uses a small guide RNA to recognize double-stranded DNA targets. For that, Cas9 firstly recognize a short sequence of DNA called PAM, and Cas9 directly interacts with this sequence. Then it opens DNA strands. One of the strands is paired with guide RNA, while another strand is displaced, and two active sites called RUFC and 18H cleave separate DNA strands to generate a double-strand break, which is what is required for double-strand DNA cleavage. And the next double-stranded DNA break are repaired by cellular repair machinery, and two most common pathways are called non-homologous end joining, and this process is error-prone and usually leads to small deletions or insertions or knockouts, it means gene inactivations. Another pathway is called exact, and it's called homologous recombination. And when homologous template are present, it could insert or delete sequences in exact position and lead to gene replacements or correction. And technology works in all kingdoms of life, including human cells, animals, plants, and bacteria. And despite successful use of Cas9, technology still has several important drawbacks. One of the drawback is limitation of target selection. This is because Cas9 target is dual. One part is recognized by of target of RNA, but another smaller part called PAM or protospace adjacent motif is recognized directly by protein, and therefore this RNA target should always be selected near the PAM. And also for most usually used Cas9, PAM is NGG, is still limits some application. For example, where exact targeting is needed, for example, base editing, or when we need to target AT-rich genomes like plants, or when we need to target only one of the two alleles. Another drawback is off targets. About it we had in previous presentation, and actually sometimes Cas9 can tolerate mismatches between target sequence and guide RNA, and this could lead to cleavage of genomic DNA in undesired locations. And last but not least, limitation is delivery. Well, to get efficient therapies, firstly, these tools needed to be delivered in as many as possible cells of the organism, and it's especially important for in vivo therapies. And currently most popular strategy to deliver vector to deliver tools are called adeno-associated viral vectors, and they have limited cargo size, and this most popular Cas9 protein did not fit in vectors, so it means smaller tools are desired by many scientists. And from previous work before establishing Cas9, we already knew that actually different Cas9s recognizes different PAM sequences, and also that different Cas9s could have different specificity, especially when they recognizes longer PAM sequences. first of my story today will be related to the characterization of Cas9 orthologs from different bacteria species. So actually, Cas9 proteins are found in almost half of bacteria, and they are naturally part of CRISPR-Cas systems. And so-called CRISPR-Cas systems confer resistance against invading nucleic acid, including phage DNA. So when the phage unknown phage injects DNA in the bacteria cell, so-called Cas proteins recognizes this sequence and inserts these new sequences into so-called CRISPR region. Then this CRISPR region is transcribed into long RNA molecule, which is further processed into small RNAs called CRISPR RNAs, and all these CRISPR RNAs possess information about different viral sequences. And finally, these Cas proteins combine with CRISPR RNA and it forms ribonucleoprotein complexes, which recognizes again invading phage sequences and then efficiently destroy these sequences. So, and in some cases, in some systems, only single protein like Cas9 is responsible both for RNA binding, guide RNA binding, viral DNA recognition and cleavage, and these systems are most interesting for gene editing applications. Actually, we started then look at these Cas9 sequences and found that actually more than 10,000 different Cas9 sequences at that time. It was, I think, 5 years ago. And we performed phylogenetic analysis, built phylogenetic tree, and selected like 100 proteins representing each clade or each family of Cas9 gene sequences from this phylogenetic tree. So what is also important that we found actually that this size of Cas9 varies from 900 to 1,500 amino acids, and as I said, these smaller proteins could be most interesting for delivery with viral particles. So then when we synthesize genes, we needed to decipher two components of Cas9 needed to get Cas9 activity, and which are specific for each of different Cas9 proteins. So first component is guide RNA. So usually we just found these sequences by performing bioinformatic analysis and then confirming experimentally. And another component is PAM sequence, which, as I explained previously, is recognized directly by protein. So far, it's impossible to predict it experimentally. Therefore, we used like library of the DNA molecules, which contains 7 randomized nucleotides in the position in the place where PAM should be located, and this allowed us to found more than 80 Cas9 proteins, which recognizes 50 novel PAM sequences, and those sequences were very diverse, and we found that actually sequences including A-rich, G-rich, T-rich, or C-rich sequences. Also, we cloned more than and characterized in vitro more than 30 of the different Cas9 orthologs, and we found also that some some proteins are very stable and retain activity in wide temperature intervals, while other proteins, like this one, could be only started to cleave DNA only at room temperature. So it means it could be used for example, if we need tighter control of the tools. And last not least, we also characterized different cleavage patterns of each enzyme. And as I said, that Cas9 has 2 active sites which cleave both DNA strands independently and usually at the same position of the DNA. And actually this most popular tool, Cas9, cleaves like DNA in opposite strands, but chops one of the DNA strands. And also we found that actually some of the tools cleave DNA, cleaves like ideally blind DNA dense, or leaves 1 nucleotide or 2 nucleotide overhangs. So this property of Cas9 also could be important because different cleavage patterns could lead to different repair outcomes within a cell. So summarizing this part, so we show that actually Cas9 orthologs have different biochemical properties and could be rich resource for potential novel genome editing tools. Also, Cas proteins, we've identified Cas proteins recognizing more than 50 new PAM sequences. Also, we show that actually some of Cas9 or Cas9 orthologs are active in broad temperature intervals, and that cleavage pattern varies between Cas9 orthologs. And this work was published in 2020 in Nature Communication paper. And also in collaboration with Kiana Ran from EuropaBio, from whom talk you already heard today, we actually found that Cas9 orthologs could be useful also for in vitro application like detection. So actually what Cardia did is they actually used one of our Cas9 orthologs called MJ Cas9 to achieve 1 nucleotide specificity and detection specificity. And actually they found that actually one of the mutations they were interested, which leads to sickle cell disease, actually is located on protospace of Cas9, therefore not in the PAM region, therefore it could not be recognized in single nucleotide specificity. However, this MJ Cas9 PAM overlapped with mutation, and this is even therefore MJ Cas9 bound only to wild type protein sequence, not mutated region, but not with mutation, and this actually led to achieve distinguish between homozygous and heterozygous forms of the mutations. Next, my story related to characterization of novel families of RNA-guided nucleases. And actually, one of Kazim's strategy is to constantly look for new available Cas protein sequences. And actually, we were lucky and identified one that actually in public databases exist near the CRISPR region protein sequence which contains only which contains RUF-C domain, which is required for DNA cleavage. However, it do not have. However, other parts of proteins do not have like domains which are similar to characterized protein. Therefore, we predicted that it could contain, could cleave double-stranded DNA despite the differences from other known Cas proteins. So firstly, we wanted to elucidate whether these proteins cleave double-stranded DNA. For this, initially we cloned this modified CRISPR system in E. coli vector, and modification included T7 promoter in front of Cas12L protein, and also CRISPR region contained spaces which were targeted our randomized PAM library, and then we used positive selection assay to detect double-stranded cleavage, and this assay includes like incubation E. coli crude cell lysate with PAM library. Then we ligate adapters to the cleaved DNA sequences, and after that amplify only by PCR only those sequences which were cleaved by RNA-guided nuclease. And if we observe if there are double-stranded DNA cleavage, we can get PCR product. So after some screening, we were lucky and found that actually found conditions where we can find double-stranded DNA cleavage. Then we sequence those PCR fragments by NGS and found that actually all 3 of the tested proteins recognized CCY PAM, which is quite novel for those so-called Cas12 proteins, and also found that actually DNA cleaved differently from Cas9 in PAM distal protospacer region. Next, we wanted to find out which components of this CRISPR system is required for double-stranded DNA cleavage. For that, we use the same positive selection assay, and initially we just removed all the sequences between Cas12L and CRISPR array. And actually, we found that then we did not get PCR product anymore. It means that some of the important sequences is encoded between in this part. And when we reconstitute actually this part between Cas12L and other so-called Cas1 protein, we found that we can detect PCR product again. And it means that most probably another RNA which required for DNA cleavage is located here. And when we took a closer look at this region, we found actually that it contains, even though only 4 DNA sequences were known at the time, we actually were able to identify some double-stranded DNA RNA structures which usually are encoded by this trace RNA. So this led us to design potential guide RNA sequences, and only one of the tested design supported DNA cleavage. Therefore, we used this design for the experiments. And when we have all purified all of the components, then we assembled the complex in vitro to confirm that actually only guide RNA is directing the cleavage of enzyme. Therefore, and actually, in these experiments, as you see, only when Cas12L was mixed with specific guide RNA, only then we observed that the band for evidence for double-stranded DNA cleavage. It's clearly demonstrated that Cas12L is RNA-guided double-stranded DNA endonuclease. And also, we bioinformatically identified amino acid which usually forms RUFC active site, and also mutation in this active site abolished fully DNA cleavage, confirming that actually RUFC domain is responsible for double-stranded DNA cleavage. Next, we looked at the cleavage pattern of enzyme. We used two techniques. One is just related to deep sequencing data, another is Sanger sequencing based, and in both cases we observed that actually Cas12L protein leaves 5' and protruding 5 to 9 length nucleotide ends. So, and this is also characteristic to other nucleases with a single RUFC active site. But when we try to test cleavage in vitro for more targets, we observed that actually only some of the target sequences were cleaved efficiently, and we still working to understand what is the reason. But actually, we found that actually when not linear but supercoiled substrate is used for the cleavage reaction, then we get like more efficient DNA cleavage. It means that most probably enzyme has some difficulties for unwinding DNA targets, because in supercoiled DNA strands are untwisted, and therefore it's easier to separate DNA strands. So since by other properties Cas12L is similar to other Cas12 proteins, so we also search for single-stranded, non-specific single-stranded DNase activity. And actually this activity is, as I said, is characteristic for many of Cas12 enzymes. Once they recognize DNA sequence, and both DNA strands are cleaved by a single RUFC DNA active site. This active site is still in the enzyme remains bound to the RNA target, and it can digest then any single-stranded DNA substrates which are in the reaction mix. And since this recognition is single turnover reaction, that it means that one enzyme is required to recognize one DNA and cleave single DNA sequences, and this reaction is multiple turnover, so it means that many single-stranded DNA sequences could be used, and this is could lead actually to signal amplification. And actually these Cas12 enzymes were successfully employed for detection of SARS-CoV-2 and other viruses, but this step still differently from CRISPR chip requires amplification of viral genomic sequences. Then this signal further could be amplified using single-strand DNA probes, which are digested by active enzyme and leads to fluorescent signal. So to test whether Cas12 also has similar activity, we use initially phage M13 single-stranded DNA, and as you see, we saw observed degradation of this M13 phage DNA only when double-stranded or single-stranded DNA sequences complementary to CRISPR repeat were present. And we next characterize this enzyme in more detail and observe that actually both short single-stranded DNA and single-stranded RNA sequences could be cleaved by Cas12L when activator is present. And last not least, we wanted to test whether Cas12L could be is active within a cell or in heterologous environment. For that, we used E. coli cells at this stage, and we actually for those we used again plasmid which encodes Cas12L and spacer against target plasmid. And when such E. coli cells were transfected with plasmids with target or control plasmids, we obtained colonies only in the case of control plasmids, which clearly demonstrates that actually one of the Cas12L proteins could cleave double-stranded DNA sequences within a cellular environment. So, and summarizing this work, we demonstrated that actually type 5, that Cas12L and enzymes are another family of type 5 proteins, and this work was published and in EMBO reports paper, and this paper were released only last week. So summarizing my work, this work, Cas9 offers great opportunities for potential exploration of Cas9 orthologs, other types of Cas proteins, also CRISPR-based molecular tools, and novel gene editing techniques. And we believe that developing new molecular tools based on CRISPR technology can benefit to many therapeutic areas. And on the final part, I would like to thank all the people which were involved in this projects, especially Cas9 team, also scientists from Corteva AgriScience, New England Biolab, Vilnius University, and also EuropaBio. So thank you for attention, and I will be happy to answer your questions.
So thank you, Gedris, very much. And maybe any questions from the audience? Yes, we have a question. And where is our microphone? Okay.
Thank you. So in principle, you have done all the experiments on probably oligos or plasmids or some kind of bigger pieces. I'm curious, I mean, if you have the methylation, you know, in relation to this cleavage of the Cas9 or other forms which you already developed and tested, how sensitive are they to these kind of cytosine or adenine methylations, you know, which are occurring, you know, in the nature, and whether you can actually change it depending also on that status of the cell?
There are no, I think, published data on effects of methylation. So most probably, if it's in this region which is paired with RNA, so most probably it's not influences the binding and cleavage. Only exception could be if methylation occur within the PAM sequence, like in this case CC PAM, but also, I think it should be tested experimentally.
Any additional questions? If no, then thank you, Gedris, very much. And the next presentation will be also online from the person in US. So we were in US before the coffee break. We came back to Lithuania, and we are moving to US again. And the presenter will be Josh Young from the Corteva AgriScience, a person who was already in some slides today. So Josh is a research scientist and fellow in the Department of Genomic Technologies within Corteva AgriScience. And with over a decade working with plant genome editing, he has amazing experience and knowledge with various genome editing platforms, including homing endonucleases, talents, CRISPR-Cas9s, and other tools. In this process, he has developed biochemical and cellular methods to assess and improve activity and specificity of genome editing reagents. Josh Young has received his BS in biology from Iowa State University and MS in entomology, focusing on the molecular or biochemical characterization of insect development proteins from University of Wisconsin-Madison. Starting as a research associate at Corteva almost 20 years ago, he has worked on a variety of projects, mostly notably transgene insertion site characterization, the generation of male sterile sorghum, rice, and wheat, and the development of genome editing tools. In the genome editing field, he has authored 17 manuscripts and over 20 patent applications. Currently, he manages teams at Corteva AgriScience and collaborates with groups at Vilnius University, Kazime, New England Biolabs, and others dedicated to establishing improved DSB reagents and approaches for genome editing. So let's see Josh's presentation.
Hello, and Lagos. So first of all, I'd like to thank the organizers, Monica and Irma, for inviting me to speak on CRISPR-Cas applications in agriculture. My name is Joshua Young. I'm a research scientist and fellow at Corteva AgriScience. Corteva stands for Heart of Nature. Launched in 2019, it merges the rich heritages of 3 different U.S. businesses, DuPont, Dow Chemical, and Pioneer Hybrid. Together, these 3 companies bring a combined 432 years of agricultural expertise and business experience. Our purpose is then to enrich the lives of those who produce and those who consume, ensuring progress then for generations to come. At Corteva, we have industry-leading R&D capabilities focused then on producing high-quality seed for row crops, as well as the protection of those crops once planted. Key functions include genomics and breeding, biotech, advanced phenotyping, data science, chemistry, discovery, and formulation, as well as the development and use of natural products to protect crops. Across this, our digital ag platform then plays a key role in helping plant the right crop on the right acre while monitoring performance during the growing season. Farmers' crops are under attack from a number of different environmental challenges. These include changing weather, drought, floods, heat waves, diseases, and pests. Additionally, we strive to make farmers successful by understanding consumer needs. This includes then our dedication to producing a healthy food supply, cleaner labels, a commitment to environmental sustainability, less food waste, and providing transparency on where products have come from. We also strive to provide market-driven, differentiated solutions to farmers. These include increased productivity and yield protection, proactive development of solutions that extend durability of traits like pest resistance, anticipation and response then to evolving regulatory concerns, meeting consumer demands and preferences, changing our and others' mindset from environmental mitigation to one developed to environmental benefit, In all, we believe that genome editing can play a central role in addressing farmers' and consumers' needs today and in the future. Genome editing is a team effort. It relies on the creation of accurate reference genomes, robust double-strand break tools, plant transformation, and thorough molecular analyses. Genome editing is a sequence-dependent technology, so having an accurate reference is a must. Since plant genomes are more often than not complex, we have partnered with BioNano and PacBio to develop state-of-the-art capabilities then in genome sequencing and reconstruction. Over the past several years, our advancements have been staggering, ultimately reducing the cost and time required for obtaining genomic sequences for plant genomes while decreasing the number of scaffolds or gaps per chromosome. This has been no easy feat, given the repetitive and polyploid nature of most plant genomes. In the genome editing space, we have developed a tool discovery, characterization, and optimization pipeline. This work leverages collaborations with Vilnius University, CASIM, and New England Biolabs. At Corteva, we have developed then an informatics pipeline to fish out existing as well as novel CRISPR nuclease diversity. Next, then in collaboration with Vilnius University, CASIM, and NEB, we have devised tools to characterize activity of new CRISPR enzymes, addressing key questions like, does it cut DNA targets? If yes, is there a protospacer adjacent motif or a PM requirement? Many of the novel reagents we discover come from diverse niches and require optimization to function in a eukaryotic cell. Using yeast or other model systems, we first introduce all possible amino acid combinations at each individual position of the protein. Those that positively impact performance are then combined together and screened for a synergistic effect. Next, improvements then are validated in crops and, in some cases, human cells. Through our work, we have discovered two new families of miniature nuclease, Cas12f and Cas12l. Here, I'll touch on Cas12f, as you have already heard about Cas12l from Idris. Its main defining feature is its miniature stature, being 2/5 the size of its larger relatives, Cas9 and Cas12a. When we first identified Cas12f back in 2017, we named this new family Cas-alpha. Since then, they have been called type 5U3 and Cas14 and are now officially classified as Cas12f. Importantly, a patent has been awarded to Corteva for one ortholog, SP Cas12f. Ultimately, this allows Corteva then to decrease its genome editing cost footprint, alleviating costs associated with the licensing and associated payments required for the use of other RNA-guided editing tools. Here is a snapshot then of some of the characterization we have done. We've established that they are RNA-guided in the presence of a 5' PAM, cut double-stranded DNA targets, and form a homodimer around one guide RNA copy. Out of the collection of Cas12f proteins initially characterized, one, SP Cas12f1, was shown to work in plants. However, it required elevated temperatures around 45 degrees C to function robustly. To remove this limitation, we then perform protein evolution using yeast as our model system. With this assay, red equates then to DNA target cleavage. As mentioned on the previous slide, the wild-type protein requires temperatures close to 45 degrees C then to cut its DNA target site. Using this red-white colorimetric screen, individual substitutions were identified that improved activity not only at 37 degrees C, but also at 45 degrees C. When combined and screened for additive effects, variants like Alt35 exhibited improved DNA target cleavage activity across all temperatures tested. With any genome editing tool, it's also important to evaluate specificity. For this, we have repurposed our yeast assay to get a quick look at guide RNA targeting specificity. By introducing single mismatch mutations into the spacer then of the guide RNA, we were surprised to find that Alt35 has a high specificity seed region that extends out to around position 18. This then can be contrasted with SpyCas9 that has then a high specificity seed region that only extends to around position 10. It should be noted that this is just a quick first look at specificity, and more thorough evaluations in plants and human cells are coming. Next, the improvements observed in yeast were validated in plant and human cells. In corn, the wild-type enzyme is active at 45 degrees C, as discussed earlier, while Alt35 shows improved activity at 45, 37, 33, and 30 degrees C. In human cells, at 37 degrees C, Alt35 exhibits targeted mutation frequencies near SpyCas9, although with considerable target-to-target variation, and is an active area for further improvement of this protein. The delivery of genome editing tools into plant cells is another critical component of the genome editing process, and Corteva has pioneered advances that utilize the morphogenic genes Wushel and BabyBoom to improve corn transformation. Functioning as transcription factors, they kickstart cell division, creating undifferentiated tissue that can be rapidly regenerated into plants. We call this transformation method quick corn. Importantly, the use of Wushel and BabyBoom genes also allow elite varieties, often recalcitrant to transformation, to be transformed with a 100% success rate. To further improve transformation, we have also modified the virulence plasmids used by agrobacterium to infect plant cells. First, 2 of the 3 plasmids then typically used were combined into one. Next, then, to prevent loss of this modified vector, a gentamicin soluble marker was also incorporated. And then finally, a better origin of replication was included, and several of the virulence genes modified. Although these advancements have led to the generation of, altogether, these advancements have led to the generation of PVIR9, which not only increases the transient delivery of the tDNA, as shown by monitoring GFP expression, but also increases the overall success rate of transformation. We have also developed a robust pipeline to evaluate the integrity of genome edits. It uses a battery of different molecular techniques, from PCR, amplicon sequencing, and a method called southern base sequencing. This approach utilizes the capture of DNA sequences used in transformation, followed by Illumina sequencing to then precisely define genome editing outcomes, for example, trait insertion, as shown in the image here. Additionally, it confirms that other transgenic DNA sequences used in transformation are not present in the final product, leaving only the genome edit behind. At Corteva, we have classified genome editing into 3 different approaches. This includes the modification of individual genes. We call this genome editing 1.0. The alteration of chromosome structure, which we call 2.0, as well as the highly multiplex genome editing approaches shown at the far right. So we then call this genome editing 3.0. First, I'll share examples of our efforts to insert and stack beneficial trait genes into plant genomes. For this, agro or agrobacterium is used to deliver our morphogenic, selectable marker, and genome editing reagents. For gene insertion, a DNA repair template containing the trait of interest destined to be inserted into the genome. And upon delivery into plant cells, the genes in the tDNA undergo expression. Once the Cas and guyronate are expressed, the repair template is excised and used as template then for the repair of a double-strand break introduced simultaneously at the genomic cut site, where successful repair results then in the seamless insertion of the gene of interest. Through our work, we have found that the release of the repair template increases the probability of obtaining plants with a targeted insertion. Indeed, the frequency of perfect and seamless gene insertion is enhanced by about 3-fold in experiments that release the repair template versus those that don't. Here's a look at how our research has translated then to product development. Dependent on target site, frequencies of successful trait insertion range from close to 2% to 11%. Additionally, trait gene packages as large as 31 kb were successfully inserted into the corn genome, although with slightly reduced efficacy. The use of our gene insertion approach also enables the stacking of multiple traits into a single complex trait locus, or CTL. The standard model utilizes random trait insertion across the genome. One can imagine that several randomly inserted traits that segregate independently during plant breeding become a monumentous task to track and combine together in the final product. Using a CTL model, downstream trait tracking and integration into elite varieties becomes much simpler when all trait genes are combined in a single locus. Additionally, using genome editing, target sites then can be pre-selected so that traits can be inserted into safe harbor locations known not to contain genes. We have also found that this approach minimizes variation in trait gene expression, sometimes observed when traits are randomly inserted using the standard model. Furthermore, since sites of insertion can be predefined, we have also chosen positions that have low sequence diversity, are away from major quantitative trait loci, or QTLs, or naturally occurring structural variants, and in regions of high recombination. Altogether, this ensures then a more rapid and smooth integration of trait genes as a single genetic stack into elite product lines. Today in corn, we have developed 4 different CTLs. So 2 on chromosome 1, 1 at the top of chromosome 3, and then the next on chromosome 10. Next, we have added another layer of flexibility when creating our CTLs. By incorporating flip recombinase target sites, or FRTs, genes inserted in CTLs can be exchanged. This allows CTL lines to be recycled and utilized then to rapidly evaluate the efficacy of different target gene combinations. Finally, we have found that different Cas9 orthologs expand targeting options with our CTLs. When selecting targets in plants for genome editing, we apply a rigorous informatics screen to select unique targets to minimize the risk for off-target cleavage. When this is overlaid with the requirement in our CTLs to avoid genic regions, Cas9 orthologs with longer PAMs have a larger number of usable sites. So ultimately, this allows us to target regions inaccessible when using SpyCas9. When tested across sites overlapping then with SpyCas9 targets, we found that other Cas9 proteins also function then to insert traits. So here, SpyCas9 frequencies range from 0.7 to 4.5%, while orthologs produce anywhere between 0.4 to 3.1% then of gene insertion, depending on the site targeted. Next, I'll describe our efforts to delete large chromosomal fragments. In the case of a QTL, pairs of targets are used together to systematically excise genes, then to determine which is responsible for the observed trait. In this experiment, we sought to excise fragments ranging in size from 220 kilobases then to over 1 megabase. In our initial experiments, we were pleased to see that frequencies of perfect excision range from 1 then to 8%. And also, it was very interesting that the size of the deletion did not seem then to impact the frequency. So, right, indeed, our largest deletion over 1 megabase was the most efficient in our initial experiments here. Finally, I'll discuss our efforts then to engineer the genomes of plants through chromosomal inversion and relocation. For chromosomal inversion, we have observed that they occur naturally across our plant germplasm, and that some of them may limit genetic gain by preventing or locking these regions from participating in recombination during plant breeding. To correct this, we have devised an approach to reinvert these chromosomal aberrations, essentially unlocking these regions for plant breeding. Using 2 CRISPR target sites flanking the inverted region, double-strand breaks are simultaneously introduced, and then through NETJ base repair mechanisms, ends of the chromosome reoriented and the chromosome reinverted. Although frequencies of perfect reinversion were low, close to 0.2%, we have shown that this approach is feasible. Optical mapping using BioNano in combination with targeted sequencing has also been used to confirm the preciseness and integrity of this chromosomal engineering approach. Lastly, we have shown that it is also possible to relocate a chromosomal region from one chromosome to another. In this case, a disease-resistant QTL on chromosome 10 identified in our tropical germplasm was relocated to chromosome 1, placing it adjacent to one of our CTL regions. So for this approach, 2 consecutive steps are required. First, the distal end of chromosome 10 is exchanged with that from chromosome 1. Second, so as not to lose any genes on chromosome 1, its distal end was added back to the now QTL containing chromosome 1. So I like to call this method chromosome dancing, and it's really amazing to imagine that this could even work, but it does. With that, I'd like to then thank all those who have contributed to this work, so including our external collaborators, Vilnius University, CASIM, and NEB. Thank you for your time.
Okay, so thanks from all of us to Josh Young, and best regards. He definitely is watching, probably now online, or will be watching the recording soon. So now we have the final presentation of today's symposium, and it will be given by Mark Guel, who came to Vilnius from beautiful Barcelona in Spain. And Mark Guel is co-founder and scientific head of Integra Therapeutics, and his interests are focused on molecular technology development and synthetic biology. And Mark's laboratory is currently centered on novel precise gene writing methodologies in mammalian genomes and skin microbiome engineering. Mark Guel is principal investigator and tenure-track professor at Pompeu Fabra University, and Mark has received multiple distinctions like European Molecular Biology Organization Young Investigator Award in 2020, National Science Award to the Young Talent by Catalan government 2018 and Spanish government 2022. He was named highly cited researcher in 2019, and so on, among others. Mark Guel has also participated in the foundations of several companies. Genesis Bio in xenotransplantation field, Biomedic in skin microbiome engineering, and of course, Integra Therapeutics in gene therapy field. So let's welcome Mark. Yes.
Hello. Well, first of all, thanks a lot for this very, very generous introduction, and also for inviting me to this symposium. I think it has been a very nice experience, and actually it feels, I don't know, very emotional to me to be in Vilnius, a place that has been so important, right, for all this CRISPR development. Very especially, yeah, and very remarkably in this point, I really like how Professor Burgess described it, right, before Cas9 and after Cas9. So definitely. Yeah, a pleasure to be here in this very important point of CRISPR development. Okay, so today I will speak mostly about one of the methodologies of mammalian gene writing that we have been working on the lab, which is based on combining the precision of Cas9 with the gene transfer capacity, in this case from a DNA donor, of an engineered piggyback transposase. So yeah, I think has been told from multiple angles today. We have this incredible, I don't know, revolution. First, in the more the genomics space, right, that we learn how to read genomes so fast and to a high throughput level that we can sequence human genomes in a few hours. But also, very remarkably, we have learned how to write DNA. So we have written already several chromosomes for both of prokaryotic, like bacteria, E. coli, mycoplasma, and even some eukaryotic chromosomes, for example, of yeast. And very importantly, also we have learned how to edit these genomes in situ, especially with this, I think, fantastic molecular machine, which is the Cas9. I think this has clearly impacted science at multiple levels. Of course, basic science. This is a number of publications that cite one of these CRISPR tools. First generation, I refer to nucleases, and then second generation, these, let's say, base editors, primators. So really it has permeated in, I think, almost all fields. I think it was very well represented today, right? We saw even like, let's say, CRISPR as a hardware, CRISPR to edit human cells, CRISPR to edit plants. So I think it's incredible to see how transformational has been. And also CRISPR is being used as a medicine. It was saluted in Professor Burgess' welcome. But yeah, so definitely these are the number of clinical trials around the world that use CRISPR. So we have had these first steps on using CRISPR as a programmable nuclease for genetic engineering, which has been extremely useful, especially in this case, at least in the mammalian context, very much exploiting this endogenous repair machinery, either in the direction of non-homologous end joining, but also in the direction of homologous recombination. Definitely this, especially in the case of non-homologous end joining, is definitely something that even made it to the clinic. I'm sure we have all heard about these ex vivo and in vivo applications of where this Cas9 protein is already used as a medicine, which is very, very exciting. Then we had this second wave of innovation where the CRISPR enzyme was complemented with other biochemical activities. This is one example of the base editors where the aminase is complementing this directioning power of CRISPR to really do direct chemistry on the nuclear on the DNA basis. So first these cytidine deaminases, so to make the C to T conversions, and then later the A to G conversions using basically this precise chemistry on this very big molecule, which is our genome. Other elements have also been developed, like combining with reverse transcriptases in the case of prime editors, and also very interesting, this natural element that have been discovered, these CRISPR integrases, which also sound very promising. So far, they have been reported to be a very effective tool for bacterial genome engineering. So yeah, so we'll see how far they can go. So then we have this quite interesting landscape and quite diversified landscape where we have the nucleases that they can trigger this DNA repair on the cells that can be used for different types of edits, especially knockouts, but also, of course, knock-ins, the base editors and prime editors, which are excellent tools to do mutation correction. And then we have these transposases, which seem to be a very good tool to do genome writing, but so far mostly in the bacterial domain. So basically, CRISPR efficiencies for gene disruption are very high. We have also base editing and prime editing for allele correction. But what happens about gene writing? What happens when we want to write large portions of genes, especially in a more generic manner, where we cannot use homologous recombination like we can do, for example, in the tissues that divide a lot? Traditionally, we have been doing gene writing with the classical vectors of AAVs or retroviral vectors, or even DNA transposases. However, these tools, while they'll have advantages that they are very efficient, sometimes they create issues on safety. Some reports have been shown that they could be linked to some episodes of oncogenesis. I have to say that not all these cases have been fully clarified, but definitely, probably in the future, we would really like to have the control where we insert payloads especially in the human genome. So then, in the case of traditional gene writing, we have these, let's say, safety concerns, but not only safety, also in terms of versatility, because very often, actually, as we could see in the second talk today, it's very useful when we can control where we insert, because then we can do a knock-in and a knockout at the same time. And then, in terms of CRISPR gene writing, so far, the efficient methods that could be demonstrated are based on HDR, and HDR, it's a very good tool, but it's a tool that works in the metabolic system, it works in T cells, but probably in other tissues we cannot exploit it. So we were wondering how we could come up with a technology that could be applicable both in dividing and not dividing tissues to transfer large payloads of DNA. In our case, so we combined what we call a fine module, which is the Cas protein, and then a cut and transfer module, which is based on an engineered transposase, which I'm going to explain in detail how it does look. And what this allows us, it allows us to find these positions in the genome and transfer the DNA payloads. This is a little bit just a very simple cartoon. So Cas9 scans, it marks the genome, and then the transposase part, it delivers this very hot DNA that has been prepared by the transposase into the genome. But the first step that we had to do when we started this project, and this came after many different attempts, it was to develop a very sensitive reporter, because actually it's not easy to detect when you get an insertion with very low background. So we try to do exome capturing, we try to do promoter capturing, but all the systems that we came across, they had very important, especially background. So one solution was to split the reporter, in this case the GFP, into parts. One part that is in the genome, so we have half GFP in the genome, and then another half that is the part that we are writing to the genome. So then we will only have fluorescent cells in case we have precise delivery where we are targeting the control, the construct. So and this, is what we call this Hershey reporter, which has been very efficient to first detect good mutants that do gene writing, and later I'll show us also how we could use this even for evolution purposes to select good gene writers. We tried different topologies of this molecular machine, so like C-terminal, N-terminal, different variants of the Cas enzyme, and we found that we could detect some, at that moment, quite a small activity, but measurable activity of gene writing when we had the C-terminal fusion of the piggyback enzyme. So and once we had this very basic activity, and actually in this case, look, so we have a small activity and also a lot of off-target. So the purple is on target and yellow off-target. So how we can improve this molecular machine to make it practical? So first we went to phylogeny. So piggyback is the same family of enzymes as the TN5 transposase, also the HIV integrase. So we did a little bit of sequence homology, and because of course from the HIV domain we know many mutations that affect the retargeting of the viral integrase. And with that, and also doing some structural studies, especially trying to check what are the most important DNA recognition residues of the piggyback transposases, we started to plan some mutants to increase the on-target and decrease the off-target. So, and here are some of the results. The summary, to cut a long story short, is that we detected that two aspects were important. One of them with excision. So basically, if we had a strong excision, we would get higher on-target activity, which is probably quite intuitive, because then we'll have more donor ready. And also to basically address the region that recognize the DNA, so that then we transfer all the responsibility of DNA recognition to Cas9. Also, we wanted to check basically if what are the biochemical features of this process. So when we separated the DNA recognition basically activity to a zinc finger, and then we were co-localizing with a separate Cas9 the double standard break, and we could see that to get high activities, at least in this version of the molecular machine of the genome writer, we need both a double standard break and a co-localization of the payload to be inserted. In the beginning, as I said, we had quite important off-target. As you can see here, we're targeting the TCR receptor. In red is all the insertions detected. In this case, most of them off-target, even though we could detect the on-target. Maybe you can see it very small here. And then after the engineering, then we could clean very much the background. And here you can see that only we detect the on-target to quite low levels. So this is with a technique that is 0.1%. Actually, today we just published this after a long review process, this method, which we call insert-seq. I'll just make a small parenthesis to explain. Actually, it was available as a bioarchive since quite a while, but from today on, it's also available from the Genome Biology as an edited article. And basically, this method, it came out from necessity in our lab to measure this precise genome writing with two features, because there are many methods to do, let's say, inverse cloning or mapping of genes, but we're really interested in developing a method that would be respectful with size, so that we could run in a nanopore machine and then not only be limited to regions that are like simple of the genome, but also be able to cover repetitive regions which are quite abundant in the human genome. So basically, the molecular biology is not very complex. Actually, it's an STAT-PCR-based approach with unimolecular barcodes, and also being very respectful with the size of the DNA, so that then we can process large pieces of DNA. And the reason being, because then we can still map even in repetitive regions, like, for example, line elements or sign elements, or even retroviral pseudoviruses. Also, one important part, which is not experimental, so the pipeline that we have made available through this paper, it will process all the reads, no matter if they can be read or not, if they can be mapped or not. So even if your of targets would map into a region, let's say a centromere, that cannot be specifically mapped, they will also be included in the report. So then you will know how many of targets you have, regardless if they are like in repetitive regions or not, which I think for therapeutic applications is quite important. Okay, then back to the gene writing. This was just a small parenthesis. Then also some more features of how this gene writing happens in our molecular machine. So one important aspect to highlight is that very often transposases, they are bidirectional. We have insertion, but also we have excision. So in our case, given that the process of insertion is destructive, it generates some, and it's not TTA dependent, so piggyback only inserts normally in TTA sites. In our case, we don't have this requirement, given that it inserts exactly where Cas9 cuts. So then the insertion is normally, but it's always irreversible. So basically, we only have, it's like an integrase, not so much as so many of the transposases. So and here we can see the motifs that are generated. So the signature, we can see, in addition of the insertion, that we have a signature of non-homologous end joining in either 5-terminal and 3-terminal of the insertion. In terms of efficiency, the efficiencies are quite reasonable, are comparable to normal transposases in most of the models that we have tested. We have tested in normal lab cell lines, muscle cell lines, neuronal cell lines, lymphoid cell lines. So it's quite portable to different systems, but from what we have seen. It's relatively easy with proper optimization to get into double-digit efficiency. And yeah, this is just some examples with a couple of cell lines, a muscle cell line, and then a lymphoid cell line. Also, we have been benchmarking this technology with other technologies, like homologous recombination or homology-independent methods. Basically, we get quite good efficiency, quite good comparables, but especially the benefit of using a gene writer that depends on a transposase, it's very apparent with long pieces. So this technology scales quite well with size, so we can deliver payloads. We have delivered up to 11 kilobases without big penalties on the efficiency. Also, we have been developing versions with increased efficiency. Piggyback acts as a tetramer, so we have seen important benefit of making dimers, so really already making a prepackaged dimer. And the reason is that very likely when we have a dimer, then the percentage of complexes that are fully integrated is higher. And also, we have been exploring other variants, which I don't have too much time to go into detail. And also, we have tried other architectures, especially using RNA interactions to bring together everything. So it's important that it's co-localized, the gene transfer machinery with the directioning machinery, but it doesn't need to be through a fusion, which sometimes is challenging, especially when we want to produce the recombinant protein. So also we can split the components, and it still works at a similar efficiency, actually even slightly higher. So, however, here, this was mostly a manual effort that we, after reading the biochemistry, studying the structure, but at the end, we're very interested in running evolution to really increase the number of variants to let nature choose the best genotypes. So what we did is we made variants, a big number of variants of most of the regions that we think were key for the activity of this molecular machine. And we ran directed evolution. So this basically was a process where we use a lentiviral platform that we can generate many variants, and then we can select these variants based on the Hershey assay that I mentioned before. Basically, this is a split GFP, so cells only turn green when the two halves come together thanks to the gene writer. So this allowed to do multiple cycles of evolution. And this is a little bit outdated. So actually we have 2 more cycles still with the trend going up, where we could improve basically the activity of this programmable transposase more and more. And actually, curiously, most of the mutations that emerged from evolution made complete sense. So actually, some of them, we had seen them, especially in the field of HIV integrase, or it made complete sense from the structure. Also, we have been trying to benchmark the system, everything I showed until now was with SP-Cas9. So we have tried to benchmark with other Cas systems. So the system seems to be quite basically portable. So we can get decent activities with multiple Cas systems. Actually, we have tested more than the ones that I'm showing here. Maybe we see some correlation with the smaller Cases. We tend to have more efficiency, but well, relatively mild correlation. And then also we have been working with ancestral versions of Cas9. This is a paper that I think it will come out soon from a collaborator. And also in this case, we could also detect some decent activity on this protein, even though this protein is not as efficient as a normal Cas9. So we are trying to polish this protein using directed evolution, but it's something that is not done yet. And here we are polishing the protein from 2 points of view. One point of view is non-homologous end joining, but also from the point of view that we are more interested, which is the basically precise gene writing using this Hershey assay, basically the reconstitution of the GFP. Another very important point for Integra is not only benchmarking this technology with the normal cell lines, but also to see that these elements can work efficiently in maybe more therapeutic tissues. So we have been working on an ex vivo program that on one side we have been producing RNP complexes. Well, not really RNP, because in this case it's RNP and D, because it's, of course, the protein, the RNA, but also the DNA that we use as a mini circle or doggy bone for delivery. So we have some preliminary results, but definitely we need to invest a bit more to optimize the processes. In parallel to this, we are quite excited about another alternative for ex vivo, which has been using lentivirus as a delivery. But in this case, not lentivirus is as the conventional use, but really VLPs, so virus-like particles, where we can load the writing machinery and then get this efficient delivery and gene writing. So the first attempts, we had moderate on-target to off-target ratios, but actually after some optimization, we could get quite important on-target. Actually, we could detect only on-target. This off-target is off-targets of Cas9, actually. So probably with a better gRNA design or better or more precise Cas, they could be cleaned out. And importantly, we can get this gene writing at a level that is not the same as a wild-type lenti, but is in the same order of magnitude, which is quite interesting. This is in Jurkat cells, so now we are trying to scale this into like T cells, real T cells. Then I'm getting to the end. So also we have been trying to demonstrate this technology in vivo. In this case, we have followed mostly a non-viral approach, even though we're also using these VLPs, but most of the work so far has been on non-viral approaches. So in the very beginning, we started using polyplexes, so basically solutions in the order of polyplexes. Now we are migrating more into LNPs. But yeah, the data I'm showing is with polyplexes. And here we are using all the reagents as RNA or the DNA as a mini circle, and then do the gene writing in this case, into the liver of mice. So in this case, it was just luciferase. We are trying to put this into context. This is more, not so much at Integra, but together with some collaborators. We have been trying to use a therapeutic model to demonstrate this technology. In this case, we have been working for some time in a type of congenital muscular dystrophy caused by mutations in the LAMA2 gene, which is a very large gene in the extracellular matrix of the muscle. We chose this disease, of course, because it's a big unmet medical need, but also because we think it's it's a good fit with our technology. So it's a large gene, cannot be delivered with conventional AAV. And then also, there is one curiosity, which despite being a muscular dystrophy, it can be addressed both ex vivo and in vivo. So we have been running 2 parallel programs, very early stage, so very early stage of program, where we are delivering basically in this case, the large LAMA2 gene into the bone marrow, where we could detect some important phenotypes, and also in vivo using the polyplex or LNP technology. In this case, we deliver to the liver. There are some thoughts that this protein can travel well, so we can detect the protein in the plasma, and now we are assessing the animals if we can also see the preclinical benefit. We have done also some tests on the germline because we believe that also this gene writing could be very useful for biomedical modeling of disease, especially because it scales well with size. So probably we could also use it for modeling of different genotypes associated with disease. In this case, we have done some germline mice, and also we are collaborating with a company in Barcelona called The Clinics, also in seeing if we can establish a method for fish to basically fish models using this technology. And yeah, so basically, just to sum up, we have been engineering these transposases to make this gene writing tool that is precise, but still relatively quite efficient. Also, I think it was very interesting to see that using directed evolution, we could improve these functionalities. And to be honest, this I think now, with all this big revolution in artificial intelligence, we could bring it even more forward. This was at the end old-fashioned directed evolution. But I think now we can bring this one step forward, coupling with AI, especially these language processing models. Also, the technology scales well with size, so definitely we can deliver in the order of 10, 11 kilobases, probably more, because these transposases have been shown to be able to deliver very large pieces, even up to 100 kilobases. Of course, then we have other problems, but at least the molecular machine can handle very large pieces. And then, yeah, we have been optimizing these reagents to establish good protocols for the relevant therapeutic models, both ex vivo and in vivo. And yeah, and then we are looking forward to next steps in seeing this technology moving to closer and closer to being useful for some therapeutic application. And yeah, and with that, I'm just closing, thanking the team in Barcelona working on this technology. And yeah, and to all of you for your attention, and happy to take the questions.
So any questions from the audience? Yes. We should just leave the microphone. The same person.
Thanks, Mark. So in principle, I mean, when we do the genome editing here, I mean, we of course expecting some kind of effects of target. I mean, but then of course there is possibility of insertion of the DNA or rearrangement of the chromosomes and other things. I'm wondering whether you actually could follow some of the pieces of the DNA, you know, which you would remove, for example, in certain cases. What the outcome of this piece, you know, which is taken away from the genome, you know, what is the future of this piece of the DNA, whether you could follow this somehow, you know, when the cell divides. And then the second question is how actually these things, you know, with genome editing could be improved if we would modulate also the cellular DNA repair mechanisms, you know, that you combine this with another one to have a better improvement, whether there is any work ongoing in this direction.
Yeah, it's a very interesting point. To be honest, regarding the first question, it results very challenging to detect rearrangements at these levels. So we are trying to expand insert-seq for rearrangements, but so far it has been quite difficult. Whereas we can get quite decent sensitivity in detecting off-targets of gene writing. However, it's very entertaining to get. There is one way that we can do a variant of the insert-seq protocol where we sequence in both directions. So insert-seq at the end, you need a primer to follow your payload. And in this case, one thing that we would suggest, if we really want to follow up these structural variants, is not only to follow the payload, but also to follow the other side of where you are targeting. And then, hopefully, we should be able to detect some of the translocations or loss of material, which is definitely a very important thing, and very likely in fastly dividing tissues, I'm pretty sure that this is something that happens, even though it's not always easy to see. So I think that's a very important point, that today, to be honest, we don't have a very, very good solution, but probably we could adapt this. The second question was about the cell state, right? And this definitely, I think it's very important. Actually, now that we got into, for example, T cells, we saw how important it is to catch the T cells at the right moment, right? Because we can go from almost zero editing to an editing that is very remarkable. In our case, especially that we depend on a double-stranded break, yeah, definitely, I think that's very important to. We are not doing this yet, but we are considering to use, for example, inhibitors of basically of the sensors of the DNA damage response. So I think that's definitely an important point. However, we need to be careful, because even that we use a mechanism that is not super dependent on DNA repair machinery, we still depend on non-homologous end joining. So probably in this case, we need to be careful when we inhibit the DNA, basically the DNA sensing to, for example, reduce the toxicity, but still leave very active the non-homologous end joining.
Any additional questions? If no, so then another round of applause for Mark. So we are slightly in front of the schedule with our second part, but we, finalized the presentations, and I will just take a couple more of your minutes. First of all, I once again want to thank first of all, the sponsors of this great event, so Thermo Fisher Scientific, Cazyme, Vilnius City Innovations Industrial Park, Research Council, as well as partners, Go Vilnius and Life Science Center, as well as the organizers, Lithuanian Biotechnology Association. And I also very much want to thank our speakers who, came here today or joined us and who really make great work, great contribution in the CRISPR and gene editing field. But actually even more, I want to thank all of you who work in this field or who are interested in these technologies. And we are all the ones who actually make a change in gene editing. So thank you all, and let's have one more round of applause for all of us. So have a great day. Thank you.